Welcome, everybody, to this week's Tech Talk. Thank you for coming. This week's talk is on adjusting parameters and differential equations with data using Python, and I think we'll get into a bit of MATLAB at the end, if I'm correct. Here we have with us Dr. Esteban Hernandez from MathStats to present. We appreciate it. Thank you for that. You have been teaching the differential equations course in mathematics and statistics, and this is a bit of an extension of that work. So I think with that, feel free to get started. If there's anything else you'd like to say.
No, I mean, if you have any question, comments, just please put it in the chat and we will try to answer them. So what I will present is very dense. I just will try to do it in a very short way. So as you see in this picture, most of the times all engineers, including myself, physics and other areas, we are trying to do mechanistic models, basically using differential equations. Most of the times, we know how to make differential equations. We develop with physics, engineering, but when we have data, we don't know how to merge data. This is a bit the future, because envisage, I guess, even all engineering. We will have hybrid models using mechanistic models, physics models, and also we will have black box models, because many things in engineering, physics, and many other fields, biology, we don't know how to model. So at the end of the day, we will merge these two models, and we should be able to train both models. But this is a little futuristic part. I will be dedicated myself only to this part, the mechanistic one. So why mechanistic? Or I call it mechanistic, everything between engineering, physics, biology, like we build based on knowledge or engineering knowledge. So if you see here, most of the times model complexity is very large. So for instance, in chemical engineering, if you want to model distillation columns, you need to make the mesh equations, that they are content of differential equations. All these equations have parameters. So most of the times we fix based on physics law, but they are based on ideal conditions that for sure doesn't happen in real life. As you know, we are getting more and more machine learning approaches. They are very good, but they are so good when you have really a lot of data there just to fit models, like large language models. But in engineering and many applications, we are not here yet. Still, our data generations of many applications are not that large. So we are oscillating here. So sometimes models in between will be benefit more if we are still using common differential equations, partial differential equations. So differential equations can go into main flavors. Most of what you study in math 310 is deterministic. So they are based on high number of molecules or particles. They also have conditions of well mixed. We assume concentrations. We don't really assume entities there. It's powerful tool because we have a lot of theory from math. We consider free noise. And every time you click, you get the same result. Stochastic is very important for many other applications because it's more realistic. We assume dilute well mixed conditions. So it's very important if we want to speak about eradication or persistence, something like deterministic cannot really do. And if we also have noise in the process, the stochastic models are very good for that. So if we go to a deterministic approach, I will define in a very general way differential equation here, dx dt. And if you see here, I am having a variable x that is changing with respect to the time, and I should have a function, and this function can be nonlinear. And x can be also a vector of different variables, it could be x1, x2, x3, x4. And if you see, I just have this general operator f. It sometimes can be linear. If it is linear, we can have some nice property that we can use linear algebra and we can work further. So here we have the conditions of why a function could be linear. But I will go more in detail what I call mechanistic model, how we build some based on general engineering laws, and later I will explain how we can build them. So in mind we have a box here. You have a component A. So in mind you have the level of the component A respect to that time. And you want to know how it's changing A respect to that time. What we could do, well, we could use conservation of mass principle. We have an input of component 8, and we take out component 8 with this A0. So if we make a mass balance, we have AI minus consumption plus production. By consumption and production, we could have there a chemical reaction producing component 8, or consuming or producing component 8. And then we take the component 8 out of the box. We can do this in very general cases. For instance, if you speak about some organs or a part of the body, you could make mass balance, and then you could change. You could be checking how certain cells are changing in certain organs. This is very common also in chemical engineering, with reactors, and even in electronic engineering, as you are having always a mass balance present there. So I will not go too much in detail in this part, just to remember like differential equations are everywhere in engineering. So here we can model how would be the consumption of component A with the first order mass action law. So we have a constant and we have a variable A. So for instance, if you assume your variable A is just separating, it's just being consumed, and you don't really care in what. So you could see how would be the dynamic of this variable A, and it's decreasing with respect to the time till it goes to 0. You always see is a parameter K, and this parameter is a whole story that we will work today. How we can find the value of those parameters K. Here, if A is transferred to B, so A is consumed, B is produced. How fast is produced, how fast is consumed A? Well, it's all based on this parameter K. Sometimes you can have consecutive fractions. A plus B gives you C. And you can see A is being consumed, doesn't go to 0. B goes to 0. Why? Because it's a limiting track. So it's a component A has the least. So A cannot be being consumed more. Why? Because I don't have B. So these are the general laws we are always learning in different parts of chemistry. This is a consecutive reaction from A to B to C. So we can see how A will be consumed, will be produced B, and then eventually B will be consumed, will be produced C. So we can go in more details of these mass action laws. I presented because they are based for many of the models we are using in different engineering fields. They are being used a lot, but sometimes we don't. For instance, they are the base for SIR models. So if you remember with coronavirus, were a lot of models how to predict the behavior of infected population. So they are based on lockable diagram models that they were proposed in the 1925, and basically are related to the problem of prey-predator. This was starting a bit before. It was an observation that they were doing in the Atlantic Sea. So basically, the fishers were trying to get fish, and sometimes they were very successful, but for some seasons, they were not able to get any fish. So they were asking a mathematician if they could figure out what was happening. So the conclusion was a big fish was eating the small fish. So as simple as you have too many small fish, then the big fish will start to eat more. You will have so many big fish that the small fish will start to die, and you will have yourself left. So if the big fish doesn't eat, what happens? They will start to die. So you will have there a periodic behavior. And it can be modeled with these two simple differential equations. Basically, you have here x is a prey, y is a predator. You could assume prey is a small fish, rabbit, whatever you want to assume in your model. So we have always a constant production of this small fish, and it's being consumed with the rate beta. And we put this one negative because it's taken out of that pool. And here is positive. We assume beta is promoting more the proliferation or production or birth of new big fish. And then this is the half-life of those big fish. In any model, including machine learning, differential equation, any type of model, a statistical model, all models are valid only under certain assumptions. So if we don't work with the model on those assumptions were based, the model basically will predict other things are not really logic to the problem. The assumptions in this problem, they are simple, but sometimes are not trivial. For instance, the prey population, fine, always plentiful. The food supply of the predator population depends only on the prey population. During this process, we are simulating the environment doesn't change in favor of one species or the genetic adaptation to the other. So, are the most simple assumptions we have there. So if we continue a bit with the system we're preparing to do parameter fitting. So here I present differential equation not in the conventional way, but with a dot. This dot implies dx dt. And this is my nonlinear function. If you see I have x as a function of t. And now I have theta. So I now start to put theta there. Theta is a vector of the parameters we want to recover. Y is the output. Sometimes in any process, you have many measurements, but maybe you cannot measure every variable you have in your program. So most of the times in biology, we only have one variable that we can measure, not that well. And we have so many other ones that we don't measure. That doesn't imply we cannot try to estimate parameters on the other variables. Is what we're going to work today. Ideally, we should have a measurement on Y as many variables as work. That one will be nice, but it's not really the case. If you see here, theta, the derivative of theta, like is a vector, is equal to 0. Equal to 0 because basically theta is assumed to be constant. In most of the problems, under certain times, certain conditions, I would say, most of them, theta is a constant. If you have a problem where theta changing respect to the time, then what I will show today to you is not going to be applied. The main assumption is like the parameter theta is constant during the time you're sampling your data. In math, we always call this problem to recover theta based on the output. We will call it the inverse problem. How is the pipeline we're going to work? Well, we will have data. We need to work a bit with preparing the data. We will formulate a mathematical model. You need to have a model you believe is going to make a good job with this data. I will not teach you how to make models, but I can tell you even a model and a set of data so you can try to fit the parameters, so you can find those parameters. We need to find a cost function or learning function, as they call it in machine learning. Basically, it's a function I will try to minimize my predictions with respect to the data. I will use the way of thinking in statistics from likelihood profile to know frequencies versus Bayesian. As I have a cost function like will be the residuals, I will try basically to reconvert this problem in an optimization problem. Then I will have parameters that you can do some model validation, some predictions, and in the engineering field, we could do some interpretation of those predictions. a bit, my life is a bit as follows. We get experimental data, usually biologists in Excel files till now. Then we start to build different mathematical models, depending on what is the type of problem, we will choose different type of model. The approach we are going to use today is not only for differential equations. It can be used for other types of mathematical models. We'll check something about identifiability analysis that I will explain later. We will check parameter uncertainty and then do some model prediction. Most of the times, I forgot to say, in engineering, if we're talking about electronic robot, hand that you want to manipulate, most of the times your predictions are very good and you're happy with your model. Depending on the engineering field you are working, if there is something related to biology, like bioengineering, or working with immunology, most of the times our predictions will fail. But that's not a big problem per se. So very likely what happened is like your model was not really, or the hypothesis you were having for that model was not right. So basically, you need to modify those hypotheses, and in consequence, you will need to modify the model. In the moment you're modifying the model and you get new experimental data, in that moment you are reformulating hypotheses, and that's generating knowledge. Hopefully, after a couple of rounds, your predictions will start to be aligned in experimental setting. I will go in with this cost function, like it's not, it's the simplest cost function that you could ever think. You have your data, your prediction minus the data. If you see, I just compute the Euclidean distance between one time in my simulation with one time point of my data. And if you see, I put to the power of 2 to the square. Why? Because I don't care if my point or my prediction is over the data, in that 10 point, or is lower. I just care that distance. And as I will care that I'm hitting all the data, so I don't care if it is positive or negative. I just care about that distance. So for the reason, it's 2 to the power of 2. If you do some statistics, for instance here, you could say, okay, if you are using the log of your output and the data, you get to the conclusion that minimizing the residual is equal to maximizing the likelihood. Like I will go and speak a bit later about it. So here I just finished the first part, a very general introduction to what is mathematical models with differential equations and the cost function that we are going to use. If you have questions till here, you can let me know. If not, we will go to the next part of the presentation. So we have a cost function that we want to optimize. So the next point will be how I will optimize it. So, and the question is not trivial because now I changed the topic from recovering parameters to an optimization problem. And I guess everybody that will do optimization will know it's a very naughty problem, like it doesn't really have a simple solution. And in the case of parameter fitting, it's very complex. So I will show today to you something about evolutionary algorithms. Why I'm using this? First, I don't use a conventional optimization algorithms based on Newton or gradient descent. Why? Because they will go to local mean. Other algorithms, there are so many, and I think I will explain that a bit here. So I am now in this part, in the optimization. and I am aiming to minimize this cost function. So evolutionary algorithms is not something new. It's very old. And sometimes I get surprised how old things are. So this is coming from 75. And of course, there's been a lot of operate in many of the algorithms. So in the 80s was a genetic algorithm, an algorithm very similar to that one I will use today. And if you go to Wikipedia, basically you can find a very large list of evolutionary algorithms, neuroevolution, genetic programming, gene expression, and I think the list is much larger. Maybe it says ant colony, this algorithm, Foucault search, the particle for optimization. So from all this huge list that all of them are promising to be very good algorithms, I choose one, and I will say why I was choosing that. I did it basically by brute force. I checked, like in my postdoc times 10 years ago, I checked all of them. And I can tell you some of them has good properties for certain type of optimization functions to work. But I found differential evolution having a very good property. It's consistent. So in the search, while it warrants global optimum, if your problem is having some differential conditions, some continuity properties, we cannot say we're finding the global minimum per se. So perhaps depending on the computational time, if you limit it, you might go just to local minimum. So we cannot certify we're finding global minimum with differential evolution. But based on different benchmarks that has been developed, not only from my side, but many others, we can find that differential evolution, generally speaking, will give a very decent job to an optimization problem. For parameter fitting, is very consistent. If you if you are using differential equations, you have a very nice property to keep continuity because there's a property of the differential equation. How it is working this algorithm? Well, it's based on the philosophy you have a seed. In mind, you have a sample that you will explore versus conventional optimization algorithms. You're always starting from one seed. And if you start very close to a local minimum, you will go to the local. Here, you start with a large seed in the whole problem. Then you start to do something I will explain. It's called selection, crossover, mutation. And after a few cycles, we basically finish the whole thing. So crossover that I have there, I mean, the other concepts are quite intuitive from evolution. Selection, we have a cost function, the cost function we want to optimize. So selection is basically based on that cost function. Those parameter values that are giving me good trajectories that minimize the distance between my simulation and the data are good sets. And just by definition, those set of parameters will be good. But I don't state just those that I was doing a good job. I do a recombination process that is called genetic information. After I will do some mutation, even they were good, and I will start to check in the selection process. So there's some wording like everything in machine learning. We have the same in statistics or math, but we call it a bit different. So individual is basically any possible solution we can have. Population is a group of all individuals. Fitness is a target function that we want to optimize. Trait is possible aspects of an individual. Genome is the chromosomes of an individual. So if we come here, so we generate the first seed in a random way. It cannot be biased, like we start to put it close to the global. The fitness of every individual in the population in each generation will be checked, and we will have a lot of potential fitness there. This is a paper. It's from Prime Store in 1997. So there are a lot of extensions of the original paper, the differential evolution. And you may say, why we're attaching to the original? We have been checking the new ones. The problem with the new ones might bring some speed in computational time. Sometimes they are faster, finding the global optimum there, but increases significantly the hyperparameters we need to fine-tune in the optimization algorithm. The original algorithm, basically, we just take it as it is in the libraries, either in Python or MATLAB. And I always suggest don't play with the hyperparameter. Because if you start to play too much with the hyperparameter, then you basically are somehow fine-tuning a lot your solutions. This one is very robust. If your problem is well posed, it will find the set of parameters that are going to give you a solution. So the original algorithm, as I said, well, is having a vector based on all the individuals. So here is just the generalization, like the xi, that can be a set of different variables I am going to use. And here, well, it's got to be those six that will serve to start to explore so many trajectories that we are going to throw there. So differential evolution, you need to give a lower bound and upper bound. And this one we need to do it. If you have a parameter, let's say the decay of T cells, or how fast can be the growth of a bacteria. So the decay cannot go to negative values. It cannot be, so my lower bound will be 0. What will be the upper bound? Well, a cell cannot die in a couple of seconds or milliseconds. So if you are not sure, you can put maybe 1 hour. The larger you put the lower bound and the upper bound will be larger the search. So in that case, it's nice, this kind of the prior concept of the Bayesian approach, that you put lower bound and upper bound based on your knowledge. But if you just put minus 1 million to 1 million, then differential evolution will struggle to find, because basically we'll discretize many solutions in that lower bound and upper bound, and we'll start to navigate. If the navigation is like the Pacific, it will take a lot of time. So it will be nice to keep this lower bound and upper bound based on reasoning of your engineering problem. If you don't know many parameters, even for me, I don't know where to put this lower bound and upper bound. Most of my parameters are positive, cannot be negative. So at least I start in 0. The upper bound, depending on the problem, I can be generous to put 500,000, 10,000. But then you can start to observe you don't need to have such large upper bound. This is the most tricky parameter to fix. This one, I don't play with it. It's called mutation. So basically, if you see differential evolution with the points or with a seed will start to generate vectors. And these vectors will be merged with other ones. So for instance, in this one, it's a hyperparameter in the algorithm. It will start with one of the best solutions after one iteration. And basically what it will do here is to recombine a solution that was the best with another. And this will bring me a new vector that I will evaluate in the next generation. Some people say this is start to bias the whole problem. You could choose brand. Basically, it's not taking the solution. You're taking random solutions in the first iteration and it start to generate new vectors. This will take a bit longer, but it's most unlikely that we'll go to local minimums versus this one. So I think both are okay. I don't think you need to play with any of these two. But if you see your solutions are not really fitting to the engineering perspective you may have, maybe you can try this other. So basically here is a bit how it will work. If you see, I will have my seeds, I will have so many vectors, and I will start to generate new vectors based on the previous two. This is a bit how it works. This is my new vector. This is the global minimum, and you can see how differential evolution basically is throwing solutions, generating vectors, but somehow with a gradient thinking, trying to find different potential solutions. The good point doesn't stay with the solutions, otherwise definitely it would go to a local minimum. What it's doing is having this point of crossover and mutation. So if my new vector that is generated by the recombining mutant vector Ig and the target vector X, so it will have the following way of thinking. I will keep this vector if the solution is less or equal than certain threshold. I will show it a bit after how this threshold works. Otherwise, I go for the target vector. I think I have it here. I think this here helps more. So this is my target vector, and we can think in the target vector like different blocks. And this is my mutant vector. So you see this one I have only diagonal lines, and this one I have diagonals in both directions. So the crossover basically is just throwing random numbers, and when it's less or equal than certain threshold, it will keep this one instead of the original. So if you see, we have a new combination of these two vectors. The original, I bring the mutant, I keep the original, I bring the mutant. This makes the whole process to be a bit slow, because at the end of the day, even we have a very good solution, we're combining it with other solutions might not be the best. Why we need to do that? Because we need to have some certainty. We don't start to bias the whole process, make a normal optimization algorithm based on Newton that will start just to follow a gradient design. The selection is a common way of thinking. I will have a solution. If my solution is better than the previous one, I keep this one. If my solution now is worse, I keep the previous one. The code, this in C. We will not do it in C. But in the original paper, this is I just copy and paste from the original paper. So you can see how the code is looking. It's very simple to program differential evolution. It's nothing obscure there that you can program in a couple of lines. We have been testing the algorithm, as I said, with different settings of the hyperparameters, and we did not observe big changes there. We will do today this example. This is done with MATLAB, and basically it's working well to follow. I mean, to find the parameters like my differential equations will be adjusting to the vector. In Python, we can use typing. It's done in a library that we can just download differential evolution. If you are using typing, you don't need to download it. We will do it today as well. And there is a tutorial of differential evolution and a benchmark there that you can see. Usually in the normal courses when I teach machine learning and I have a bit more time, I start to show differential evolution, how it can find global minimums from very complicated functions in math that we know what is the output. So it's how we make the benchmark. We know the solutions. And we start to test if differential evolution is finding really the general, the global minimum. So this one is a very famous function, the McCormick function. And there are so many other ones. So I will, at this point,
So I have a question.
yeah, sure, tell me.
So, your genome that you use, in your evolutionary algorithm is entirely just a vector made up of the parameters to your differential data.
Yes,
There's
exactly.
nothing else in there, it's just the parameter.
The parameters yeah, exactly. The parameters will start to make different vectors. I mean, if you have 10 parameters, your space will be large. Let's think on one parameter. That parameter can generate a solution of your differential equation. So this is the trajectory that I will compare with the data. What you have done basically is that. Thank you. So if somebody online has a question, just let me know. I put in the chat the code. I think you should see it. And for instance, we can try to open, maybe sometimes we need to copy and paste the link.
Yeah.
So that they can see. So I put some code there. As you can see my screen, I was putting differential evolution. This just to check it with normal optimization functions. Lotta Volterra. Fisher is a partial differential equation. And also, I will try to show it with MATLAB. As I know in engineering, you use a lot of MATLAB. Well, I still use a lot of MATLAB. So if you try to open the code 5.1, it's a very simple script. I have it here. I hope I don't have issues with Python. It's a new computer and a new Python version. So if you see here, I'm just importing from SciPy differential evolution. There's nothing new here. I will start to click. This one is a normal, very calculus 1 toy equation. You have the exponential minus x minus parentheses x minus 10 to the power of 4. If you make a derivative as we used to do it in calculus 1, you could find a solution. So for this one, we don't need to have differential evolution. It's to use a very powerful algorithm for such a simple problem. So let's say I don't know what is the solution. But I only will give bounds from minus 10 to 10. And if you see, trying to click minus 10. If you see, the way of using differential evolution is very simple. F is a function I'm trying to minimize, simple task. And here I put the lower bound and the upper bound. I was very generous, 100, 200. I don't have more minimum. It's a very simple task. What is doing it well? It's 9.9. If you do the derivative, you will find it's basically 10. And then if you evaluate a function, you will get this minus 1. The McCormick function that I was telling you before is a bit more complicated. It has some periodic solutions. So it's a bit more challenging to study. And you can start to simulate. If you go to the McCormick function, I think I was telling here. And if I put here the solutions, let me see. That was Then look with the screen. Something here. Mm, Well, I will not put here, but you can Google the McCormick function, and you will find basically the resolutions of this function. Mm, at minus 0.54 minus 1.5. And the evaluation of these two x to give you your y will be minus 1.91.
So
So
the function of two parameters.
two variables. These are just optimization. I mean, I have in this case, I have a 3D surface, and I want to find my minimum. I think it's in here, of x and y. It's here. But it doesn't have only one solution. It has several. So based on this, it should be here. Here is another minimum. So it's exploring the whole landscape. But yes, this is not a scale parameter estimation. If we go to the other slides. I will explain a bit, or we did a module in Python. I will explain in a bit. Even if you don't know how to use Python, I find it very intuitive. I learned Python when I was old, meaning after 13. So if you are under 30, you should be easily using Python. And I will explain some general definitions that we will need to use for explaining better the parameters that we're finding. So just as a math problem, but also in engineering, you would like to obtain parameters that are unique. So imagine you have any device, a reactor, a robot. So we believe these parameters cannot be changing just values. Why? Because it's an electronic device. So this electronic device is well settled. And in certain time, 2 years, 3 years, we don't have a significant change for maybe some months in this device. So the state of the art is like these parameters are constant. And if we have data, we're interested like that when we recover this parameter, we obtain the same one. Not like every time we do parameter fitting, oh, I obtain 5, next time 50. If I do it again, 1,000. So if you are obtaining two different parameters every time you are checking or the algorithm is not being consistent, it's because you have a problem that in math we would call it identifiability problem. I know in machine learning sometimes they are not really interested in this because you have a neural network is so huge. Just the point of tracking the parameters of the neural network, it would be challenging. And sometimes the neurons can be changing their task in doing a part of the identification. But in our case, no, we are not changing. We have fixed differential equations. They will do the same task. They are deterministic differential equations. The only way we change trajectories of these differential equations is like we change the parameters or the initial conditions. So you can assume like you don't know the initial conditions and you can say, if I want to estimate it, that is possible. But given initial conditions and a set of parameters, you will obtain the same trajectory. So identifiability basically means that theta, the vector of parameters that you will obtain, is constant and is unique. It will be global depending on the space that you are working, or will be local if you start to assume certain neighborhoods in your parameter value. There is some nice math, but I wouldn't go in detail. It's called algebraic identifiability. Sometimes it's not the problem your data. Sometimes it's like if I have certain variable that I'm observing, let's say temperature in a reactor, and I want to obtain all the parameters in concentration, pressure, value. You might not manage to do it. Why? Because it's simple. For this output, I cannot see the other parameters. Simply, I cannot see it. It's how it's based differential equations and the map to your output. But as I said, I will not talk on the math of this, but if you are interested, you can send me an email. We can discuss about it. Computationally speaking, I will show you how we can observe algebraic identifiability. For instance, in this problem, you see three differential equations. Tt, let's say a target cell can be infected with a virus. So target cell with a virus will be infected with this rate beta. Beta comes here, it's infected cells. So I am producing infected cells minus the clearance of infected cells. They will be cleared with a parameter tilde. Virus will be produced with a parameter T, is a normal replication rate, and the clearance of the virus. If we do this algebraic identifiability, we will obtain some, in one point, an equation like this. Like this is a first derivative of the virus. In this problem, my only variable I can measure is a virus. I cannot measure target cell, I cannot measure infected cell. But if I do this math, I can observe, well, you can recover delta C, delta plus C, theta is going to appear in delta C, delta plus C. It's basically telling me what is in this map, what could be appearing for that value. If using that equation, I don't have P. Basically, what it's trying to tell me, I don't care how nice is your data with the virus, you will not be able to recover P. And if you see, P is appearing in the equation of the virus, however, I cannot recover that parameter. I can't recover beta, I can't recover delta in a combined way with C, but I cannot recover P. So sometimes, even for the same variable, you cannot recover a parameter with that measure. We will show it, and I think might be more intuitive in this one. Is what we will do computationally. So imagine you have a parameter theta. This is your residuals, your cost function. So if I start to make variations on that parameter, and I let the algorithm estimate the other ones, if I obtain something like this, we may all agree this is a minimum or something over here, and we can recover that parameter back based on this field. These fields are called profile likelihood. What happened in this one? If you say, whatever I modify by parameter theta, the cost function doesn't change. So basically, you can do whatever you want, but your output, how you are modifying this parameter, is not affected. So that parameter basically is called structural not identifiable. How is your parameter with your equation, with the output you measure, you cannot recover. And the last one, if you see you have a parabola that is opening, in this case, kind of in a big way to the right, sometimes can be to the left. So basically, the data start to be too messed up. You have a lot of variation in the data, especially in biology. Your samples can go from 10 to 1,000. This will start to affect your profile livelihood. So basically, will be some moments, not perhaps in this one, but like I cannot recover the value of theta either because your space, the data, non sampling is not enough. Sometimes we will need to do bootstrapping, but I will talk a bit about that. Bootstrapping at the end of the day is to do some Monte Carlo sampling. We will set some inputs, we will generate the outputs respectively, and we will check how these parameters are being evaluated. So I would pass maybe to this figure, then we can start to play with the code. So we will generate figures like this basically scatter plots. So we want to know if alpha and gamma are parameters. So we want to know how is the distribution of these parameters based on the data we have, if there are some correlations between the parameters. As I said, for this we did a library, well not a library, a module in Python. We call it PD params. It was originally designed for partial differential equations. It can be also used for ordinary differential equations. The function is simple, so you just bring the module that I will tell you what you can find it. You need to tell what is the data, the model, the initial conditions, the lower bound, upper bound, and I will wrap it in details in these other ones once we do the simulation. You can find the original library, well, module, and all the examples I will show to you in Python. You can find it in GitHub, and you just need to go there, or this one is later. Or if you don't want to go to GitHub, you can find in the chat, there is a link in Dropbox. You simply can download all the files, and we will try to simulate them now. I will finish the simulations with a biofractor in MATLAB. So this is another example I have that you cannot find in GitHub, but I know many of you are using MATLAB. And in this example, we will have a biofractor that is producing bioplastic, and we want to basically do parameter fitting. That eventually you can imagine what you can do. If you have a model that is already fine-tuned, then you can try to optimize your process. You cannot try to aim to produce more bioplastic, reduce cost in the production, etc., etc. But first, you need to have a model that you can somehow believe on it. This is example I will work after. Any questions? So if not, I think I finished with the presentation, and then we can open the code. So here, I would ask you to open the now code 5.2, local Volterra. If you see, we have a file in Excel also called the same. Here is the file where I have my data. It's basically something. So if you see, it's a file, I have 4 columns. No, sorry, I have 3 columns. The first column is the time. If you see how I have time repeated, so every time the time is repeated is because it's a replication in your data. So I have 3 samples for 0.07, 0.07, whatever time units, and I have 3 different samples. So time, usually we need time because it's a differential equation. I will talk about when you have a space for partial differential equations, but you also will need time there. So time goes in the first column. In the second column, I have a variable that I will explain. And in the third column, I have another variable. So if you did write I will not have problems with this. So I will import pandas as PD, numpy as NP, and matplot library as PLT. Is there? Here is the module. I was having an error. Now I only have warnings. If you have the file, is here. If your file, you should have in the file that you are executing, like is a code 5.2, in the same folder, you should have the library that is called PDE params. It's the only thing you need to do. Or if you are working in Google Colab, just upload the code. Well, you also need to have the data, lockable tetra, and you need to have this module together. I am having warnings. We did this module, I think in 2018, 19. So there are many things in Python changing. So it's basically telling me some syntaxes is not valid, but I hope this is not going to affect us today. It's just some warnings that things in some of the other modules we were using are not consistent anymore. But we will double check everything is working right. So I just have warnings. Here is the, if you click, basically you can see here, I'm using later to make in a nice way the two differential equations I'm going to work. This is a predatory prey model. So I have X and I have Y. So you see it's a simple model. I have how many parameters? I have A, B, and that's it. I only have two parameters. I have two variables, two parameters, and it's a nonlinear equation, not that difficult. So I need to put this in a way Python will understand. So I will make a definition of this differential equation. I will put it in this way. X and Y equal to 6. So here I'm putting the first differential equation. As you see, it's very, let me see if I can make it here. This is exactly like this, and this is like this. I'm just doing a definition of the differential equation. You need to provide the two variables that in this context is X and Y, time, and the two parameters, A and B. Looks like it's working. We need to have initial conditions for these two differential equations. Here you define the initial condition. So I just click. And the next step, we need to bring the data. So just remember the Excel file. Well, it was not Excel file, it was .csv file. I will bring it in this way. So at the moment, everything is working. I have my three replications. I have this variable and this variable. From your data, you need to know how what columns means. The first column, I don't know if I will explain it later now. Here is time. This is my X variable, and this is my Y variable, based on the data. The most difficult part is like how we will assign this X to the X of the differential equation and the Y to the Y of the differential equation. Well, for this, we have the module, and we make some rules on the module. So the main rule we have in this module is The way that you organize here in your differential equation, x and y, if you see here, I'm providing in that order versus x and then is y, is the order it should be provided on the data. If you think it's easier, if it's too difficult to change the order in your data, just change the order in this part of your differential equation. It's the only thing you need to satisfy. The order is given in this way, x and y. So, if we go to the main module, df is the data that I have here now. I make all this in the variable df. Lotka Volterra is the function I was telling you. And here I have the initial conditions for x, the initial condition for y. I need to put lower bound, upper bound for my first parameter, for the second parameter. You can play with this, it should be consistent. Here is what I will call the parameters. I will call my first parameter A and the second parameter B. Somehow this is important because it will be the input I will be giving here. My differential evolution will throw seeds, and those seeds will be fitting in the differential equation in A and B. So if you change here the name of the variable of the parameter, just be consistent when you do it here down. How many variables I will be choosing for this optimization? I'm saying 2. Doesn't need to be 2. I will do it only with 1 after. But let's assume I know x and I know y. How many dimensions? This is not a partial differential equation, so I don't have any dimensions like you could have in a partial differential equation in x, y, and z. So we put 0. How many replicates? I know in this data set I have 3 replicates for each of the variables. And I will put, this one, if we have a nonlinear function as the output of the variables, I just will put none to this, and it should hopefully work. It worked, looks like. And I'm getting, no, it did not work like this. Here is what it's going to work. Here I just define a model. Here I define the initial conditions. And here, as any instruction in Python, I have my model. I need to do fitting of that model. Okay, this is the work. I get 2.99, 1.0. I know these parameters are right because I generated the data with this parameter. I just introduced some noise. So it's a way to double check my algorithm is doing the right thing. The error I'm having, well, it's very little, is 10 to the minus 5. I want to check a bit the likelihood, how it's looking. I will show you to you here. Just to have your model, you just need to put dot likelihood profile, and automatically, if it is still valid my module, error widget. I think it's something, well, this is still running, and it's always telling me error widget. Looks like it's finished. Yeah, I need to update my mod, I guess. Here is what is going to be the plotting. So, well, it's just generating the data, and here is what is your plotting. So you see for parameter A, you have a very nice parabola, and this is what is your minimum. I am just bringing the value differential evolution brought here. In this one, you also have a minimum. One side is opening, but still you can find your optimum is there. And now I will do bootstrapping. Why I need to do bootstrapping? Well, if you have replicates of your data, very likely you should have, even in a biofractal, I think in bioengineering you always do two replicates. So you need to see how much the variability of your data is affecting your parameter. So for this, bootstrapping basically is doing random sampling of the data and trying to do parameter fitting. So it's not taking the whole, just some sampling in a random way. So in order to do that, you just need to use the model and just put dot bootstrap. And I think I am having this error displayed widget, but technically it should be working. I think by definition it's 100 sampling. If you want to have more, you can put between the parentheses 1000 and we'll make 1000 for you. And here are the statistics. So what is saying the average of 100 samples is 2.99, for B is 0.99, the standard deviation is 0.09, it's very small, and 0.007. And if you want to compute confidence interval, computational time, it was done in 5 seconds. And this is a bootstrap summary, just to see it in a nice way. But basically, yeah. If you want to see the scatter plots, just a different way of observing your parameter. You can see the distribution of your parameter here and here, and the correlation between A and B. You can see it also here. We don't have honest correlation. So basically, you have two variables. You can recover both parameters. Any question? Okay, what would happen, and this is this example, if you only have one observable value? Let's say I only can see predator. I cannot, well, for the prey, the predator, there are too many, I cannot measure, or I don't have a way to measure one of them. So in this case, I will play a bit with my Excel file. So I will basically delete that column 1, and I just will keep time and column 2. So this one you need to remember that column 2 was white. Otherwise we will make a mistake. So here X, which we're ignoring X, we're only working with white. The main tricky point, it will be exactly the same. It's the same differential equation. It's the same initial conditions. I will not modify anything. The only thing I need to modify is here. I am working with two variables, but I'm only observing from the data the variable 1. The variable 1 is in the order of Python is working. X is 0, Y is 1. So this might be the most confusing part. Is everything well documented in the module? Just be careful what you are telling to the module is really the variable you have. Otherwise, you will estimate apples with bananas and it will not work. So just be careful, you assign the right variable there. So if you see this is a more challenging problem, I'm recovering both parameters, but only with one variable. The parameters will not change, are the same like before, but we want to observe how is the effect of not having one of those variables. So I will start just to click. If you see, I think I got stuck somewhere. Let me see. It's not that close to 3 as it was before. Now I have 2.89. And this is depending how picky is your field. So if you are in engineering working with electronic devices, I think this start to be a big error. If you are working with biology, it's nothing. So it's basically subjective to your field. If you want to check more about those parameters, you can check the error you are getting. If you see here, it increases. And I will check the livelihood profile because I want to figure out what is happening. Later, I will show you the statistics. So what is happening if I only observe one variable now? Very likely my both parameters start to be correlated because I only can observe one variable, one data set, and I want to recover again both parameters. So if I just go here, you can start to see that the livelihood profile start to be not that nice. So I start to have a line. Some, I don't know if it is just the granularity that I'm doing it. I think I just jumped too many times. I need to click first here. And then yes. Um, I think something like jump is not finding. Um, for the partial differential equation simulated, I just will show you the results because that one takes a lot of time. This is still a simple example. Everything is working. Yes, it's working. Everything. Something is working not well. I don't know what is my preference. Everything was working to this point. But that's like let me let me start to do the statistics. Meanwhile, mm, I think I have a problem with LaTeX. Mhm. Yeah. I don't have it installed in my computer. Uh, If you install LaTeX, it should work. It's basically the error. But I don't know why it gave it to me now because before it was working. Because basically I'm using the nomenclature of LaTeX. So, but now it's complaining in this one, and it did not complain before. The statistics, if I go to the statistics, you can see the standard deviation for this case really is much larger. It's 0.4, before it was 0.0 something for A. For B, it's still good. Here we can see basically the distributions are start to have a bit and a small modality there. But this one is very different than before. If you see, A and B start to be highly correlated. And this is another big problem. It's always in doing inverse problems, even in machine learning. If we start to have the data that is not really the right one, parameters will start to be highly correlated. If I modify one parameter, will affect immediately the other. The only way to solve this, bring me the other variable. It's still bringing you, in my view, relatively okay, the value for A and B, but it's all depending on how much error you can handle in your program. So as I said, in biology, I can handle a lot. Here, just it's the same example, but just assuming like my observable variable for my data is a multiplication of x and y. I wouldn't go in more detail of this. I mean, you can do it. I can show you, I would like to show you the PDE. The PDE, I will not model it. It's a Fisher problem here. I will not simulate it, sorry, because that one really takes a lot of time. So the idea is the same, but now I have a partial differential equation. It's a classic Fisher differential equation. You have your parameter, like it's a diffusion, second order with one variable in x, and you. Here, it will be everything consistent as we were doing before. However, the main difference, I mean, in the data, you will have time. We only have one variable, but we need to have the dimension. The order of the data is t, x, and u. So if we start to do it, I will show you here. If I want to find the diffusion parameter, you will run everything the same, and you will discover the right parameter. For the other parameter, when you start to have this opening, you can see from the statistics, the mean, the standard deviation is really small. This one is only one partial differential equation. But if you have now another dimension, I think I have an example in GitHub for the TensorFlow. So you have TensorFlow in X and Y. You have now two dimensions and time. That one starts to be much more complicated. Even just to simulate a partial differential equation is a whole research topic. So with this file, basically you will see the simplest partial differential equation. I think in many of your applications, at least in the courses, we at least work with one partial differential equation order. I know if you are really having a prototype, you will start to have more variables. It can handle it, but maybe you need to ask Luca that helps you to use a cluster because this will really start to take a lot of the computational time. The procedure I have is exactly the same. So I would like to go, if you don't have any questions here, I would like to open MATLAB. So for MATLAB, I only have one example, and you will maybe not be that happy. So if you open in Dropbox, this one actually, all these files, I really use it for a research problem I am doing of biofractors. So you need to go to the file that say main fitting. And it will open. It's taking time. So the main fitting looks like this. Well, let me bring also the other interface. Um, That's missing one window. I don't know what it is. So you open main file. Okay. this one. And the way of working, you need to have, but I think I have the library already inside of the files. It's a library that was done by somebody. I tested a lot. So I can tell you it's working, it's relatively okay. If we go to here, it has many flags. I mean, you need to have all these files because it was on like that, the library for differential evolution. I think the main component, I don't know if it's here. No. This is this is the main file for differential evolution. But if you take any away of these other ones, sometimes will give you an error. Somehow it's calling other files. So might be annoying to have all these files there. So I would suggest you, just try to ignore the other ones. Come to the main fitting. And here in main fitting, we need to have the objective function that we want to handle. The nice part in MATLAB, I can show you really more details because this code basically, I was doing it in my old times. So we did not make a fancy module. With a module, we don't see too many details. It's very easy to use, but you don't see details. Here we can see more details. These are the parameters I want to optimize. I have one, two, well, I want to not optimize. 1, 2, 3, 4, 5, 6 parameters. If we go back a bit to the example, just to recapitulate a bit. The example we are working, I have 4 differential equations here, and I want to recover K1, K2, I want to recover mu S, and K4 and K5 from these 4 differential equations. So I need to put lower bound and upper bound. If you see, sometimes I am very generous. Something different, like I have from Python. Here you can see a number. This is a quantization. If you remember differential evolution, at the end of the day, is just doing a quantization from the lower bound to the upper bound. The level of granularity you will do it, in this case, is 0.1. Here is 0.01. I just decided to do it less because the lower bound and upper bound is just much smaller. I think that what is here are many of the hyperparameters, so as I tell you, I don't really touch it. Maybe the only one you need to keep in mind is this one, that time of searching. This case is time of optimization. I have here only 60 seconds because I cannot stay here 1 hour to show you that result. But if you think, usually if you have at least 3, 4 parameters, you need to give some reasonable time, some minutes to search on the space of these 5 variables. If you have 5 parameters, you are looking in a space of 5 variables, in 5 space. So that one we cannot even imagine. So it will take time. You cannot just put 60 seconds. Here is a maximum number of iterations. I mean, if you put 6,000 on 10,000, it's fine. It will stop either this, or if you put a timing. I don't use this one. The way of calling differential evolution is the params. What is the params? I go back here. So the objective function I want to optimize is in objective to handle. Let me see what I call it. It's here. The params is all these settings that is done by the is what I call hyperparameters, but we shouldn't play too much. I'm just using mainly what it was in the library. And other things, actually, I don't even touch it. But you can even say, if you want to send the results to your email, that is nice. And maybe we should go to the objective function to handle. If you see, I am addressing this to another file that is called objective function fits that I will try to open for you. Because I know it's not that intuitive. Trying to be opening here. So here is basically giving back the residuals. So you have the data. I just put the data here. You don't need to put the data here. I mean, this is a very simple example. You can call a file and just make like in Python, this file equal to certain variable, and then call the different variables down. But here I have time of experiments. These are 72 hours. This is real data, by the way. 0, 4, 8, 10, 24, 72. And I have concentration for ammonium, glucosa, this variable like is a biomass, and the pHA, which is basically bioplastic. And we can see how is the production of bioplastic there. Here, I just need the initial conditions. My initial conditions, I just take it from the data because it's the time zero. So I don't need to estimate it, it's coming from the data. Here, I do some interpolation. You don't need to do this, but I found it very simple when I combine with to minimize the residue. Basically, what I'm doing is from time zero to the last point, 72, I start to make interpolations. You may say, why do I need to do that? Because when it's coming, the simulation time, it will basically try to align with any point. If you have, maybe we'll show it here. It might be easier. So so that part, what it's doing, let's say from here to here, you see the distance is kind of large. And your algorithm is going to minimize the distance between this point and this point. But what is in between is a bit obscure. So in order to make it a bit nicer, this part doesn't need to be what I do sometimes, is to provide artificial or input data. What would be the data from here to here, assuming it's linear. So basically, I am giving more data from here to here in this way. You don't need to do it. You only can give, if you are having enough data, you don't need to do this. So the only point you need to be careful is when you make the cost function. The cost function is here. So at the end of the day, I am not estimating all parameters. I have some of them fits based on the experiments that they just passed to me. And I just want to recover other parameters. So the other parameters that I am estimating is mu, K4, KPS, K5, K2, and these are my differential equations. So basically, it will be millions of seeds trying to explore different parameters. And here is where I will start to minimize the distance between the simulation and the data. And this is given in this part. So in this case, I have 4 because I have 4 variables. If you have more variables with data, you will need to increase here the part, another sum because we are having another variable. And here is the number of variables you're trying to minimize. So you see, this one is not that automatized as it was in Python, but it's still, you can adapt this code to your problem. Just try to understand this part. So if we go back to the main file, that is this one, if you click to run, it should run. And let me see MATLAB. It's running. So this one is nice. You can see how it's evolution of the parameters in every iteration, what MATLAB is trying to search on them till the time of exploring is finished or is converging. It can converge, but usually if you have more than 3, 4 parameters, it will be complicated. So we are having 39 seconds. If you have well posed your problem, even you don't give too much simulation time, MATLAB will give you a reasonable result. So it's almost finishing, 53. This is the last set of parameters I get. 91.28, 10 to the minus 3, and so on. And technically, it's also going to throw to you how it's looking. Well, in this case, it's not that promising. Maybe I should give more simulation time. I cannot print over the screen here. Just one. It's in my other screen and I cannot pull it down. Maybe I will share other screen. But if I share screen 1, oh, I'm trapped now with the screen. On Zoom, I share screen 1, I think. So you can see the plots, but here, anyway, so you are all in Zoom. So you can see basically that it was not that good, the parameter fitting. So I would speculate it was just not enough simulation time. I will go back to the other file, to the other screen, and I will just increase the simulation time. It's not a simple problem. What would happen if you give very little simulation time as I'm doing it MATLAB, because I don't want to spend too much time. But believe me, if you give 1 hour, it will be doing a good job. So what it's doing, okay, it was exploring the whole landscape, but you are asking me to stop. So I'm just throwing to you back the research I was doing. But it's not that good, and I should connect it to the electricity, or I will not have a computer. So even if you are running, it's finished. It's still searching. So many things can affect. For instance, I am having some, I am doing other things. I found at least in MATLAB, this can affect a lot. And on the top, I'm not giving enough simulation time. So I can really decrease the search of 6 and it will give you a better result. So I would hope with 2 minutes, even I'm using some, it will try to make a good job. If you have questions till now, you can do it. Meanwhile, hopefully MATLAB is finished. This problem is more challenging than I did with Python as well. I am searching for 5 parameters. So it's very large, the search. So now it's finished. Now it's doing a good job. I will share. I don't know why I cannot bring the other screen, but I will share my other. You can see now. So now you can see many of the I mean, at least this line is trying to cut the trend. And sometimes in the, especially with differential equations, it will not have perhaps the freedom to follow every point. This is what you need to keep in mind. I'm not saying this set of parameters is the best. I mean, we observe is following. You will need to do also some bootstrapping just to see the confidence interval in these parameters. But I mean, it will be very difficult, for instance, what is my mouse, that the model goes here and goes down. Somehow the exponential growth of the bacteria is here having linear and then it's growing in different ways. Here is following mainly the last points versus the first ones. Something perhaps in my model is missing to represent better the behavior the first hours versus the other hours. So if you think you were given enough simulation time and you have all the variables, perhaps you need to modify your model. That is not really the best model to represent this data. If you are very sure this model is right and your data is right, I would suggest you increase the computational time. However, perhaps still you might not be able to estimate the full parameters. So this one you can find it with a liability profile. But it's a simple way to try to recover parameters, differential equation, and data. So this is all what I wanted to present.